For a medication to be stable and easy to manufacture, the raw material must behave predictably. We target a specific state, perfectly uniform, spherical granules that flow freely, avoiding sticky clumps or static charged powders that stall production lines. This state is achieved through drying, the process of removing a liquid, usually water, from a wet product via evaporation or sublimation. It serves as the critical final step in manufacturing before a drug reaches its packaging. The pursuit of a completely bone-dry product is a common fallacy. Stripping all moisture from a pharmaceutical powder introduces static charge inhibition, which paralyzes powder flow and causes particles to cling to machinery rather than flowing through it. Overdrying also ruins tablet compactability. For a tablet press to successfully compress granules into durable pills, engineers must target a precise residual moisture, typically held between 1 and 2%. Drying is a complex kinetic progression. Reaching that narrow moisture window requires managing vapor pressure gradients and internal capillary forces as the material shifts through different physical states. To map this progression, we must measure moisture accurately. Because the total weight of a batch continuously changes as water evaporates, using total mass as a denominator creates a moving target that complicates kinetic equations. Industry standards instead use the dry basis equation. This measures the mass of moisture against the fixed mass of the bone-dry solid. This invariant denominator allows us to track moisture loss with mathematical precision. Total moisture is categorized into two thermodynamic states. The first is free moisture, which can be evaporated under standard conditions. The second is the equilibrium moisture content, or EMC. The EMC is the steady-state moisture locked within the solid structure. It is impossible to remove through evaporation alone unless the ambient temperature and humidity are fundamentally altered. By anchoring our calculations to the dry solid mass and targeting the floor of the EMC, we can accurately model the kinetics of water removal from any pharmaceutical batch. We visualize this process using a kinetic drying curve, plotting the rate of drying against the moisture content. We read this graph from right to left, tracking the material as it moves from wet to dry. The process begins in the constant rate period. Here, unbound water covers the surface of the solid. As this surface water evaporates, it is immediately replenished by liquid drawn up through the pores from the bed below. During this phase, the rate of evaporation is identical to that of a free liquid surface. The solid's internal structure has no impact on the drying speed. The liquid behaves as if it were an open pool of water. This rate is governed by the mass transfer equation. It is determined by the partial pressure of water vapor at the surface minus the partial pressure in the surrounding airstream. If the boundary layer of air above the bed becomes saturated, the pressure gradient collapses and evaporation stops. To maintain drying, the vapor must be removed by turbulent air, or the bed must be heated to raise the surface vapor pressure. However, excessive heat introduces the risk of evaporative cooling. If too much latent heat is transferred without efficient vapor removal, the local temperature may drop to the dew point, causing water to condense directly back onto the product. The constant rate period is a purely thermodynamic stage. Success relies entirely on heat transfer and the effective removal of the saturated air boundary layer. As the surface water is depleted, the rate of vaporization begins to fall. The liquid can no longer move to the surface fast enough to saturate the air immediately adjacent to the bed. We call this threshold the critical moisture content, or CMC. It marks the point where the constant rate period ends and the drying rate begins to drop. The kinetic bottleneck has shifted. Drying is no longer limited by how much vapor the air can absorb, but by the physical transport of liquid from the core of the material to its surface. The bed has entered the funicular state. Air now occupies the internal pores of the bed, and the remaining moisture exists as thin, continuous films clinging to the granules. Within these pores, surface tension creates a capillary suction potential. This force draws the water films through the internal interstices towards the surface of the bed. Once the CMC is breached, the dryer's efficiency depends entirely on the microscopic capillary network within the material. During the first falling rate period, this capillary movement creates a significant engineering failure mode. As moisture travels to the surface, it carries dissolved active pharmaceutical ingredients with it. This is intergranular solute migration. Capillary action drags the dissolved drug from the interior of the bed and deposits it on the surface granules. As the water reaches the surface and evaporates, the non-volatile drug is left behind, stranded at the boundary between the solid and the air. This surface enrichment destroys dosage uniformity. One layer of the batch becomes overconcentrated, while the interior is depleted of active drug, causing the final tablets to fail quality control. Increasing the temperature often worsens the problem. High heat drives faster moisture movement, triggering more aggressive solute migration before the granules can dry. The effort to remove a volatile solvent can inadvertently alter the spatial architecture of the non-volatile active drug. Eventually, the moisture volume becomes too low to maintain continuous films, and the capillary network snaps. This is the pendular state. On the drying curve, this represents the second falling right period. The moisture is now held only in isolated, lens-like rings at the contact points between particles. Bulk liquid movement is now impossible. The plane of vaporization retreats into the body of the solid, and drying slows to a crawl. At this point, moisture removal relies on the molecular diffusion of vapor through the tortuous pores of the bed, a process that is significantly slower than capillary flow. Pushing a product into the pendular state requires high thermal energy for very little mass removal. This stage carries the highest risk for thermolabile compounds, which may degrade under prolonged heat exposure. Traditional compartment dryers hold material in static, packed beds. These systems rely on slow capillary action to bring moisture to the surface, making them highly susceptible to uneven drying and solute migration. Halting solute migration requires a method that severs the internal capillary network while simultaneously stripping away the saturated air boundary layer. This is achieved through fluidization. High-velocity air is forced upward through a perforated base until the upward draw of particles exactly matches the force of gravity. The bed enters a boiling phase. Massive turbulence suspends the granules in a stream of hot air, treating each particle as an independent unit. Because the particles are physically separated and moving randomly, the continuous capillary channels between them are severed. This makes intergranular solute migration impossible. The 360-degree air-to-particle contact maximizes the mass transfer coefficient. This strips away vapor instantly and minimizes the time spent in the constant rate period. The fluidized bed dryer outperforms static trays by structurally altering the physical state of the particle bed, bypassing the kinetic limitations of capillary transport. To dry pharmaceutical materials effectively, engineers must match specific heat and mass transfer mechanisms to the material's kinetic phase. Fluidized beds provide turbulence for granules, spray dryers handle liquid droplets, and freeze dryers manage thermolabile biologicals via sublimation in a vacuum. Despite these divergent architectures, the underlying thermodynamic requirements remain identical. Success depends on controlling the vapor boundary layer, managing internal capillary forces, and precisely targeting the equilibrium moisture content. Successful pharmaceutical drying relies on the control of kinetic phases to preserve the chemical and physical integrity of the medicine.